Okay, everyone. Today we're going to talk about viruses and prions. This is the chapter 5 in the textbook. And again, you may have noticed the last few classes we've sort of been taking a survey through microorganisms. We did prokaryotes. The last chapter was on eukaryotic microorganisms. And now we're going to talk about viruses and prions in chapter 5 here. Okay, so let's define a few terms. What is a virus? Now remember, everyone, and we talked about this back in chapter 1, viruses are acellular particles, They're not cells. And because a cell is the smallest thing that is itself alive, the smallest thing that can be considered living, viruses are not truly alive. They lack cytoplasm, cell organelles, and plasma membranes. And they are incapable of performing their own metabolism independently of any other organism. So viruses can be referred to as obligate intracellular parasites, which means they must invade a host cell, invade the cell of another living organism, in order to become active and reproduce. Viruses are hugely plentiful. There are at least 10 to the 31 viruses on Earth. That's 1 followed by 31 zeros. If stacked end to end, they would be 100 times the diameter of our galaxy. That's just mind-boggling to think about. And it's estimated that they invade a microbe host 10 trillion times a second around the world. And that would be mostly bacteria. Think about that, 10 trillion times a second around the world. Now, as I just said, many viruses infect bacteria. These are called bacteriophages, or sometimes just shortened to phage or phages. Interestingly, some viruses infect other viruses. These are called virophages. These are viral parasites of larger, what they call giant viruses that they co-infect. And they rely on that co-infected virus to help them reproduce inside a host cell. Now, you may remember that the last 2 chapters with prokaryotes and eukaryotes, I always said that they're mostly beneficial or highly beneficial. That's not true for viruses. In nature, viruses are parasites that cause disease or death. But there is some side benefits to that. They kill a lot of bacteria, especially in the oceans. They help regulate bacterial populations. And it's estimated that the weight of all the viruses in the oceans is equivalent to the weight of 75 million blue whales. It's about 200 megatons. And about half, 50% of all bacteria in the oceans die every day due to viral infection. So from the bacteria's perspective, it's good that they can also reproduce quickly. Another side benefit of viruses is they are used clinically and for genetics research. They're used to make many vaccines, and they're used to introduce DNA or RNA into an organism. They're a good way of transferring that genetic material, and they can be used in gene therapy. So that's a little side benefit. Now, viruses are extremely small size. I can't overstate how small they are. So if you kind of get a little perspective here, here's a red blood cell. This is a bacterial cell, E. coli right here. And right there, that's a virus. And here's another virus, and these little tiny things are even smaller viruses. So if we zoom in on that, you can see how tiny those are. It's incredibly small, okay? Here again, we see that, all right? Red blood cell, just a portion of it shown here. E. coli here. Look at how small these viruses are. Extremely. Here's images of the COVID-19 coronavirus. Maybe this one here, class of 2020, see? classic COVID. Now, when were viruses discovered? The person credited with discovering viruses is called Dmitry Ivanovsky in 1892, pictured here. He was working with tobacco mosaic virus. He noticed that these plants were infected by something. He was able to determine that it wasn't bacteria, but what was it? Is it a fluid? Is it some other kind of particle? He didn't really know what it was and just referred to it as Contagium vivium fluidium. Fast forward to 1935, and this man, Wendell Stanley, identified and observed the tobacco mosaic virus pictured here for the first time. Why was he able to do it? He used a new piece of technology called an electron microscope. Viruses are too small to be seen with a standard microscope, a light microscope. You have to use an electron microscope to view them. They're that small. All right? Now, let's look a little bit more deeper at the morphology of viruses. Now, remember, everyone, morphology refers to body, shape, or form. Shape or form. Now, all viruses, and this is important now, have two basic parts. They have genetic material, so DNA or RNA, but not both. They also have, surrounding this DNA or RNA, they have a protein coat called a capsid. So a protein coat called a capsid contains DNA or RNA. That's it. That's a virus. Now, some viruses have a third part called an envelope. The envelope is kind of an oily, semi-gelatinous layer on the outside of the capsid, similar to a bacterial cell's capsule. Now, not all viruses have that. Some, like COVID and the flu virus, do. Here are some viral shapes. See, this one has that envelope on the outside. Here are some more. Look at the shape here. All this stuff here, the little spikes and the little green things, is all part of the capsid. There's the DNA in there. This one looks like something that would land on the moon. This is a bacteriophage that infects viruses. Here's some more. This one here, this influenza virus, has the envelope on the outside. All right, here's some more viruses, just to give you the idea of shape. Now, what about the viral genetic material? Remember, viruses can be DNA or RNA, but they don't contain both, like living organisms do. Now, this DNA or RNA can be single-stranded or double-stranded. Well, you could have single-stranded DNA, double-stranded DNA, single-stranded RNA, and double-stranded RNA. And furthermore, whether it's DNA or RNA, the genetic material can be in a single piece or broken up into multiple pieces. So from a taxonomy perspective, scientists classify viruses first on whether they're DNA or RNA, and then you can go from there. So you see here, just this little thing right here, that's the entire DNA or RNA of a virus. Very, very small. All this stringy stuff is just part of the genome, the genetic material of E. coli. All right. Now, the capsid, the protein coat, is made up of little subunits called capsomeres. So like in those pictures, there's little green spheres that made up the capsid are capsomeres. They can come in a variety of shapes, as you saw in the pictures. Helical, which kind of looks like a cylinder. Each blue circle is a capsomere. The whole capsid, there's my DNA or RNA up here. It can be polyhedral, which has more of a geometric shape. And it can be complex, which would be something like this. Even more elaborate in shape. Now, if a virus has an envelope, this is that semi-gelatinous layer on the outside of the capsule. Again, it's not found in all viruses. So this one has the envelope here on the outside. There's my capsid, and there's my DNA or RNA. All right. Now, viruses that have an envelope don't make the envelope themselves. It forms from the host cell's plasma membrane. So when a virus leaves a cell after it infects it, it takes some of the plasma membrane with it, and that becomes the virus's envelope. So here's the virus here. As it leaves the cell, it takes some of the membrane with it, and that becomes its envelope. Here are some human diseases that are caused by viruses. You can take a look at that, but let's talk about how viruses work. All right? Now, viral growth is very, very different from the growth of bacteria or yeast or archaea or eukaryotic cells or any other thing, because viruses absolutely 100% need a host in which to replicate. They use the host cell's enzymes, ribosomes, and other materials to make copies of themselves, which we call viral replication. The virus's genetic material has its own genes. So once the virus gets into a host cell, its own genes can become active and make its own enzymes. But it relies on that host cell to get started. Replication usually kills the host cell. So again, a virus can be called an obligate intracellular parasite. So step one is the virus has to enter a living organism. Now remember, everyone, before they get into a living organism, viruses are not living. They're just particles. All right? So this dormant virus has to somehow get into a living organism. This can happen in a variety of ways, but viruses can't seek out a host. Like other parasites, like a tick or a leech or lice, they can seek out a host. They can go looking for a host in which to help them live. Viruses can't seek out a host because they're not living. They just have to get into a host somehow. Through the air, through food or water, through a cut, somehow. Now, once the virus gets into a host, a living organism, it can attack the host's cells, and it can make new viruses inside those cells. It can replicate new viral particles. Now, there's two basic ways a virus can do this. First, we have what's called lytic infection. Now, we call it lytic infection because at the end of this process, the host cell will rupture, will burst. A fancy name for rupturing or bursting is lysis. To lyse, L-Y-S-E, is to rupture or burst. So it ends in the rupturing of the host cell, causing host cell death. So there are 5 steps that the virus must follow. It first has to attach to the host cell, which is called attachment or adsorption. Then its DNA or RNA enters the host cell. It takes over the host cell's machinery and begins making new viral particles. Those particles are then put together, and then all the new ones, the new viruses, burst out of the cell. So attachment or adsorption, entry, synthesis, assembly, release. How many new viruses you produce is called the burst size, and that's going to vary by the type of virus. It can be in the hundreds or thousands. How long this 5-step process takes is called burst time, and that also varies by the type of virus. Now, the best way to understand this is to look at a picture here. So here we see the lytic cycle. So start here, step 1. Now, here's a bacterial cell. This is one of those bacteriophages. Step 1, it's going to attach to the outside. We see here, attachment. Now this, in purple, is the bacterial cell's DNA, its chromosome. Step 2, entry. Notice the virus here injects, kind of like a syringe, it injects its genetic material into the host cell. Now, in step 3, the first thing that this virus's DNA does, notice the capsid just stays on the outside. It's now called a ghost. This viral DNA chops up the bacterial cell's DNA, and it now takes over the cell. It commandeers the cell. Once it chops this up, it starts making new viral pieces. This is called synthesis, and then these new pieces are assembled in this order down here. We see assembly. All right? Once they're all assembled, they burst out of the host cell, and they can now infect neighboring cells, and this release will kill the host cell. Now, besides lytic infection, you can have lysogenic infection. Now, in this case, it's similar to lytic infection, but instead, the viral genetic material integrates itself into the host cell's genome. So instead of immediately chopping up the host cell's DNA, it instead inserts itself in there. Once it's inserted into the host cell's genome, we call it a prophage. And now it can remain dormant. So the virus remains dormant inside the host cells and does not cause disease right away. In fact, it can remain dormant for long periods of time. And because it has integrated itself in the host cell's DNA, if that host cell divides by, say, mitosis or binary fission like bacteria, the viral DNA is copied along with it into the daughter cells. Now, that can go on for a long time, even years. Eventually, we reach a stage called induction. Now, during induction, something happens environmentally or internally that triggers that dormant virus into an active state. The virus now chops up the host cell's DNA and takes over the cell. And remember how I said all the viral DNA is copied in the daughter cells? They all induce at the same time. So if we look at this diagram here now, attachment and entry are the same. But notice now we're going to go this way. Instead of chopping up the host cell's DNA and taking over right away, look what happens. This viral DNA, see it right here? It has inserted itself into the host cell's genome. We now call it a prophage in here. All right? Now, if this bacterial cell divides by binary fission, one cell into two, notice the viral DNA gets copied along with it in all these daughter cells. Now, in induction, something happens that triggers this prophage out of a dormant state and into an active state. All right? Now it chops up the DNA, takes over the cell, assembles, and releases. So you can see them side by side here. This circle would be lytic. We'd go over here, integration, okay, and go this way for lysogenic. All right? Now, we just looked at how that works in bacteria. What about animal and plant or eukaryotic viruses? Well, without getting too specific here, animal and plant viruses work similar to what we just looked at. In acute viral infection, that's similar to lytic, okay, similar to the lytic cycle, like chickenpox or COVID are lytic. Latent viral infection is similar to lysogenic. A latent animal virus can often integrate into the host cell's genome, just like the lysogenic cycle, like what HIV does. So chickenpox acute, later latent, shingles appearing later, latent herpes Get the reoccurrence here in 2/3 of people. All right, so it's similar. Now, if all that's not bad enough, viruses have also been linked to cancer. Some viruses, in fact quite a few now, are known to cause cancer. These are called oncoviruses. Oncoviruses. And about 20 to 25% of all human cancers are caused by viruses. Oncoviruses work by disrupting the cell cycle and causing cells to start to divide uncontrollably. So you end up getting a tumor, uncontrolled cell growth. Okay? Examples include papillomavirus, hepatitis virus, and viruses that cause these conditions here. Burkitt's lymphoma, Hodgkin's disease, Kaposi sarcoma, and others. Viruses can be difficult to work with in the lab. In the lab, we can culture viruses, but in order for them to work and grow, we have to have a host. We can use bacteria as the host, and if we grow them in bacteria, viruses form what are called plaques. We see in this picture here, this agar plate is totally covered in bacteria. The clear areas are areas where a bacteriophage, a virus, has killed the bacteria. Those are the plaques. You can grow viruses in plants, animals, embryonated chicken eggs, so a fertilized chicken egg, and other media like cell culture. Now, related to viruses, we have a category of particles called viroids. Viroids are infectious RNA. There's no protein coat, so no capsid and no envelope. Just RNA by itself that can cause a disease. So far that we know of, viroids only cause several plant diseases. Here are some images of the infectious RNA. You see what it does to the leaf of this plant here and how it stunts the growth. This is normal, this is viroid infected. All right, and that brings us to our next major topic here in this chapter, which is prions. Prions are infectious proteins. So no DNA or RNA, just a protein that by itself that causes an infection. They were discovered by this man, Stanley Prusiner, in 1982. He was working with a disease called scrapie, which is found in sheep, causing a fatal degenerative disease of the nervous system. You can see what it's doing here to this poor sheep. In 2010, he would go on to some fame. Now, how do prions work? Prions work by disrupting normal brain proteins. That's an important word, disrupting normal brain protein. So if we label a normal brain protein, PRPC, they disrupt that normal brain protein in mammals and humans. So viroids are only known to infect plants. Prions infect mammals and humans. So the prion protein, which we could label here, PRPSC, causes normal proteins to refold. So they change shape, which causes death in brain cells. And that's what's shown here. You don't have to know the details here, but the prion protein disrupts that normal protein, causing it to refold, thereby changing shape, leading to death of the cell, and particularly brain cells. The red indicates where the prions are in this brain tissue. Here, this is a normal brain. This is one damaged by a prion. You can see the difference. These clear spots there's no brain tissue there. The prion has killed it. Mad cow disease is likely caused, well, it is caused by a prion. Now, this cow here doesn't actually have mad cow disease, but it looked like a mad cow. So I like to use it for the picture. So what about prion diseases? All prion diseases are characterized by what's called TSC, transmissible spongiform encephalopathy. This is a progressive brain degeneration. It causes dementia, loss of coordination, ataxia, and inevitably death. Prion diseases, everybody, even in humans, are inevitably fatal. There's really no treatment for them. They're almost always fatal, inevitably fatal. Okay? So all prion diseases are characterized by this TSC. Now, in animals, a couple important ones, we mentioned scrapie, first documented about 300 years ago, seen in sheep 3 to 5 years old, as shown here. You might have heard of chronic wasting disease, which mainly affects deer. And you can see what it's done to this poor deer here. The yellow and red dots on this map indicate where we have seen cases of chronic wasting disease. So you'll notice there are some here in New York State. And then lastly is mad cow disease, known formally as bovine spongiform encephalopathy, or BSE, mad cow disease, pictured here in these poor cows. This was first observed in 1986 in the United Kingdom, and it's peaked. Its worst outbreak of it was back in 1993, and around that time, with about 1,000 cases per month. Now, what about human prion diseases? Probably the best known is Creutzfeldt-Jakob disease. This is a neurodegenerative brain disease. This is that picture I showed you before, normal and one affected by Creutzfeldt-Jakob disease. Look at the damage that prion does. The duration, it lasts about 4 to 6 months, inevitably leading to death. It can be inherited genetically, or it can be acquired from infected human tissue, the cornea or brain tissue. Variant Creutzfeldt-Jakob disease, first described in 1996, again in the United Kingdom. This was documented from people eating beef from those cows that were infected with mad cow disease, and it led to this variant. Lasts a little bit longer, up to 14 months, inevitably leading to death. And then the last one is something called fatal familia insomnia, which causes progressively worsening insomnia, hallucinations, dementia, that TSE, and then death. Again, this one tends to run in families as well. Okay? All right, everyone. So that's an overview of viruses and prions. We've now surveyed most of the microbial groups that we're going to be focusing on. Prokaryotes like bacteria and archaea, eukaryotes like protists, protozoans and algae, and fungi, mushrooms, molds, and yeast, and now viruses, viroids, and prions. Next time, we're going to be looking more at the physiology of microbes. So what's happening inside the cell and kind of how microbes work and what do they do. And I'm going to talk a little bit about microbial nutrition and growth. But that's it for today. Until next time, have a nice rest of the day, and I will talk to you later. Bye-bye.